Welcome to today's deep dive. We're so glad you're joining us.
Yeah, really excited to dig into this one.
Today, we are taking you behind the scenes of something you interact with all the time.
Probably every day.
Right. But almost certainly something you never think about when you do it.
Which is usually a sign that the system is working, honestly.
Exactly. We're looking at an internal FDA document today, specifically a text titled Guide to Inspections Validation of Cleaning Processes.
I know. It sounds like incredibly dry regulatory reading.
It really does.
But our mission today is to explore the hidden, surprisingly dramatic world of how pharmaceutical companies actually prove their factory equipment is safe.
Because ensuring your medicine isn't cross-contaminated, it isn't just a matter of running some vats through a giant dishwasher.
No, not at all.
It's a massive, highly complex scientific undertaking.
It's a monumental task. I mean, think about it. When you pick up a prescription from the pharmacy and swallow a pill, you take it completely for granted that the capsule only contains the medicine listed on the label.
You just trust it.
Right. You trust it. But the journey to guarantee that purity is filled with microscopic detective work, rigid protocols, and, as we'll see, a history of some pretty alarming close calls.
Close calls that totally force the industry to change its ways.
Exactly.
Okay, let's unpack this. Because to really understand the standards we have today, we have to look back at the historical context. The FDA expecting pharmaceutical companies to have clean equipment is obviously not a new concept.
No, it goes way back.
Right. Our source document notes that the 1963 Good Manufacturing Practice Regulations and the later 1978 regulations, they explicitly stated that equipment had to be maintained in a clean and orderly manner.
But historically, FDA investigators were mostly just looking for what the text calls gross insanitation.
Gross insanitation.
Yeah, they were looking for visibly inadequate cleaning.
Right, like poor dust control systems.
Yeah, or major glaring issues like cross-contaminating regular everyday drugs with penicillin.
So in those early days, the focus was largely on the obvious, visible problems.
Very much so. If a facility was making potent steroids or hormones, the FDA wanted to make sure those weren't just visibly getting mixed into other batches.
They were looking for the big, obvious failures in hygiene.
Right. But as manufacturing became infinitely more complex and global supply chains grew larger, the definition of what constituted a cleaning failure had to evolve.
Because a visual inspection just wasn't going to cut it anymore.
Not even close.
And a major catalyst for that evolution happened in 1988.
Yeah, this is a wild case.
There was a massive recall of a finished drug product called cholestyramine resin USP. And the source text shares a shocking detail about this recall.
It's terrifying, honestly.
The bulk pharmaceutical chemical used to make this medicine had become contaminated with intermediates and degradants from the production of agricultural pesticides.
Agricultural pesticides.
We are talking about pesticides ending up in a medicine meant for human consumption.
And just to add a bit of context to the name here, the USP part stands for United States Pharmacopoeia, which basically just means the drug is supposed to meet official, highly rigorous public standards for quality and purity.
So finding agricultural chemicals in a USP product is basically a worst-case scenario.
Absolute worst case. And the way it happened is this fascinating chain reaction.
It wasn't just a spill on a factory floor, was it?
No, not a spill. The contamination was traced back to a complete lack of control over reused solvent drums.
Wow.
The facility was taking drums that had previously been used to store recovered solvents from a pesticide production process.
Okay.
And they used those exact same drums to store solvents for the resin manufacturing process.
They just reused pesticide drums without thinking about the invisible chemical residue left behind in the metal.
The firm had completely inadequate controls over the drums. They didn't properly test the drum solvents, and crucially, they had absolutely no validated cleaning procedures for those barrels.
They just assumed they were empty enough.
Right. They looked empty.
The domino effect from that one assumption is staggering.
It really is.
Because this contaminated bulk pharmaceutical didn't just stay at that first facility.
No, it traveled.
Shipments of it were supplied to a second, totally different facility at a different location for finishing.
And that's where the pesticide contamination spread to the bags used in that second facility's fluid bed dryers.
Which is a notoriously difficult piece of equipment to manage.
Oh, incredibly difficult. For those trying to visualize it, a fluid bed dryer bag is essentially a giant industrial-sized porous sack.
Sort of like a massive heavy-duty vacuum cleaner bag.
Exactly, or a hot air balloon. Hot air is blown up through the bottom to dry, wet pharmaceutical powders suspended in the air.
And because it's porous and made of fabric.
Any chemical contaminant blowing through it gets deeply embedded in the material.
And because the contaminant was embedded in those bags, it ruined subsequent lots of medicine produced at that second site.
Ruined all of it.
The craziest part for you listening is that this second site was a place where no pesticides were normally produced.
Not at all.
The contamination just hitchhiked across the supply chain.
If we connect this to the bigger picture, you can see why the FDA realized they had to step up their game.
You cannot rely on a factory looking visibly clean if the invisible chemical residue from a completely different industry is hitching a ride in a reused barrel.
Exactly. And this wasn't an isolated incident either. The source text brings up another major case, a 1992 FDA import alert on a foreign bulk pharmaceutical manufacturer.
I was looking at that case. This facility was manufacturing highly potent steroid products alongside non-steroidal products.
Right.
And they were using the exact same common equipment for both. They just shared the vats.
And the FDA considered that multi-use facility a massive, serious health risk to the public.
But the firm actually did have a cleaning validation program in place, didn't they?
They did, but the FDA deemed it entirely inadequate. The fatal flaw was their testing methodology.
How so?
The facility was only looking for evidence of the absence of the previous active compound. They completely ignored the presence of reaction byproducts and degradants.
Which is such a massive oversight. Because when investigators actually tested the rinse water from the equipment, they found those exact residues and byproducts hiding right there.
Right there in the water.
The source notes they used thin layer chromatography or TLC tests to find it.
TLC is a brilliant analytical tool.
How did it work?
Think of it like taking a black marker, putting a dot of ink on a paper towel, and dipping the end of the towel in water.
Okay.
As the water travels up the paper, the black ink separates into the hidden reds, blues, and yellows that actually make it up.
Oh, wow.
TLC does that for chemical compounds. It separates a mixture into its individual components, creating a sort of chemical fingerprint.
So the FDA investigators used this technique on the rinse water and immediately saw the hidden degradants the company had missed.
Exactly. The fingerprint was right there.
So what does this all mean? At what point does a massive industrial piece of pharmaceutical equipment actually become clean?
That is the million-dollar question.
Because the FDA doesn't just want to know that a company washed it. They want scientific proof.
Yes.
But the source text outlines very different rules for different scenarios.
Right. The FDA demands that firms establish written standard operating procedures, or SOPs, for their cleaning processes. And the text makes a vital distinction here to answer your question.
What's the distinction?
If a manufacturer is cleaning equipment between batches of the exact same product, say they're running a 3-week campaign making millions of the exact same painkiller, the equipment generally only needs to meet a criteria of being visibly clean.
Visibly clean. Okay.
Between batch cleaning for the identical product doesn't require complex chemical validation.
That makes sense. If a tiny microscopic trace of a painkiller gets into the next batch of the exact same painkiller, it isn't a cross-contamination crisis.
Right. It is just more of the same medicine.
But the moment that factory switches from making the painkiller to making a blood pressure medication on that identical machine,
the rules change entirely. Cleaning between different products requires rigorous written validation protocols.
it's a whole different ballgame.
Exactly. The procedures have to be highly specific. If a firm uses one process for removing water-soluble residues and a totally different chemical process for non-water-soluble residues, their written procedures must explicitly address both scenarios.
They have to dictate exactly when each is used.
Down to the letter.
I'm curious about the really hard stuff to clean. The FDA text specifically calls out processes that leave behind tarry or gummy residues.
Oh, those are a nightmare.
When a medicine is basically a sticky paste, how do they even scrub that out of a giant industrial vat?
Often they don't even try.
Really?
Yeah. In cases with tarry and gummy residues, or with notoriously difficult equipment like those fluid bed dryer bags we talked about, firms often decide it is simply not worth the risk of cross-contamination.
What do they do?
Instead of trying to validate an impossible, wildly expensive cleaning process, they will simply dedicate that specific equipment to a single product forever.
Wow, they just buy a new machine for the new drug.
It solves the chemical mixing problem completely.
That's fascinating. But even if the residue is easy to wash away, there is another invisible enemy lurking in the equipment that the text highlights.
Time and microbes.
Yes. What happens if a factory processes a drug on a Friday and doesn't get around to washing the vat until Monday.
The text is incredibly strict about that exact scenario. The length of time between the end of processing the drug and the actual cleaning step must be strictly controlled and identified in the documentation.
Because if it dries,
If residues are allowed to sit and dry out, especially things like topical creams and liquid suspensions, they become exponentially harder to remove.
it is exactly like cooking oatmeal, leaving the dirty pot on the stove overnight and then trying to scrub it the next morning.
It turns into cement.
Literally cement.
But the microbial aspect of that delay goes even deeper. The text strictly states that under no circumstances should stagnant water be allowed to remain in equipment after cleaning.
None at all.
Equipment must be completely dried before storage. Stagnant water is an open invitation for microbial proliferation.
Now, a lot of people might assume that since pharmaceutical equipment is sterilized before the next batch is made, a little bit of bacteria in some leftover water wouldn't matter.
Right. They think the heat will just handle it.
The sterilization would just kill it all.
That is a highly dangerous assumption.
Why?
Control of the bioburden through adequate cleaning and drying is essential because subsequent sterilization procedures might not be enough to keep you safe.
Even industrial sterilization.
Even then, the source text explicitly warns about the control of pyrogens.
Pyrogens.
Pyrogens are fever-inducing substances that are often produced by bacteria as a byproduct of their life cycle.
Oh.
Even if your high-heat sterilization process completely kills the live bacteria, it might not achieve significant inactivation or removal of the pyrogens they left behind.
So you're basically leaving behind the toxic corpses of the bacteria.
Yeah.
And the sterilization doesn't remove them.
That is the danger. If the equipment wasn't properly dried and stored, the subsequent sterilization will not save the next batch of medicine from causing severe fever reactions in patients.
That is deeply unsettling. But it really highlights the stakes of getting this right.
It really does.
So how do FDA investigators and pharmaceutical companies actually test if the equipment is clean? The source text gives us a fantastic analogy to explain this.
The dirty pot analogy.
Yes, we just talked about oatmeal, but the text uses it as a core principle for sampling.
It is the absolute best way to conceptualize the problem of sampling.
Because when you're evaluating a dirty pot at home, especially one with dried out, stuck-on residue, you don't just fill it with water, swish it around, dump the water out, and then look at the water to see if the pot is clean.
No, you look at the actual pot.
Right, you look at the pot.
That brings us to the two acceptable sampling methods the FDA recognizes for validating cleaning. The first is what you just described, looking directly at the pot. This is called direct surface sampling, and it is usually done by swabbing the equipment.
Literally taking a swab to it.
Exactly. The major advantage of direct swabbing is that you can specifically target the hardest to clean areas. An operator can reach into the specific crevices that are reasonably accessible and physically scrub off dried out or insoluble residues to test them in a lab.
But factories are full of complex machinery. You cannot stick a cotton swab down the inside of a 100-foot stainless steel pipe.
Which is where the second method comes into play. Rinse samples.
Rinse sample.
Rinse samples allow a company to evaluate a much larger surface area, including those long, inaccessible piping systems that cannot be routinely disassembled for swabbing.
You run a highly purified solvent or water through the system and test what comes out the other end.
Exactly.
But going back to our dirty pot analogy, rinse samples have a massive blind spot.
They do.
If the chemical contaminant is physically glued to the side of the pipe, or if it isn't soluble in the water you are using to rinse it, the water will flow right past it.
It won't pick it up at all.
You will test the rinse water, get a perfectly clean result, and have absolutely no idea that the pipe is still coated in dried residue.
And the FDA is very aware of that blind spot. They are incredibly strict that when you test rinse water, you have to do a direct, specific measurement for the actual contaminant you are trying to clean.
You can't just guess.
Right. A company cannot just test the rinse water to see if it meets basic water quality tests and call it a day.
Here's where it gets really interesting. The text warns that a negative test result, meaning the lab didn't find any drug residue on the swab or in the rinse, might not actually mean the equipment is clean.
No, it might just mean your testing method is terrible.
Right. The FDA expects firms to actively challenge their own analytical methods.
They have to prove it works. A company has to prove that if a contaminant is intentionally placed on the equipment surface, their swab or their rinse can actually recover it.
They have to calculate the recovery rate.
Exactly. If you put 100 micrograms of chemical on the steel, does the swab pick up 90 micrograms or only 50?
You have to prove the test actually works in the real world before you can make any conclusions based on a negative result.
And sometimes the tools used to run the test actually sabotage the test itself.
Yes, the text points out a fascinating quirk about direct swabbing. Sometimes the sampling material actively interferes with the data.
It's wild.
For example, the adhesive used to hold the cotton onto the stick of the swab has been found to interfere with the chemical analysis of the samples.
It is a brilliant detail. It means that early in the validation program, chemists have to rigorously test their own swabs and extraction solvents.
Just to make sure they aren't the problem.
Right, to assure the sampling medium itself isn't going to ruin the entire multi-million-dollar validation study by introducing its own weird chemical signals.
The design of the factory equipment plays a massive role in whether a company can even win this game of microscopic hide and seek.
Oh, absolutely.
The FDA text specifically calls out large systems that use semi-automatic or fully automatic clean-in-place systems, known as CIP systems.
CIP systems.
Instead of taking the machines apart, they run cleaning chemicals through the closed loop.
And CIP systems represent a significant area of regulatory concern. The engineering choices made when building the factory dictate how clean it can get.
The text notes that sanitary-type piping should always be used.
Right. However, it is surprisingly common in the bulk drug industry to use non-sanitary ball valves.
For anyone unfamiliar, a ball valve is essentially a sphere inside a pipe with a hole through the middle.
Think of it like a tunnel through a solid ball.
Right. When the hole lines up with the pipe, fluid flows. When you turn the sphere 90 degrees, it blocks the flow.
But all those little internal mechanisms.
Yes, the gaps between the sphere and the housing. They give chemical residues a perfect place to hide when the valve is turning.
When those non-sanitary ball valves are used, the cleaning process becomes significantly more difficult. The FDA expects the cleaning operators to have special training and experience in cleaning those specific difficult valves.
They have to know exactly what they're doing.
The valves and piping even need to be physically tagged and easily identifiable on flowcharts.
Because if you don't know it's there,
you won't clean it right. The text actually notes that inadequately identified valves have literally led to incorrect cleaning practices because operators just didn't know what they were dealing with.
Let's look at some of the pitfalls companies fall into when trying to prove their systems are clean.
There are a few big ones.
Let's say a company comes up with what sounds like a genius, labor-saving idea. They say, instead of swabbing and doing all this chemical math, let's just run a fake batch of medicine, a placebo batch made of inert powder, through the dirty equipment using the exact same operating parameters.
Okay.
Then we will just test the placebo powder. If the placebo comes out clean, the equipment is clean.
What's fascinating here is that the FDA has documented fundamental structural flaws with this placebo testing method.
It sounds completely logical at first glance.
It does, but it breaks down entirely under scientific scrutiny.
Why doesn't it work in practice?
Contaminants do not wear off the equipment uniformly. Okay, imagine the discharge valve or the chute of a giant industrial blender is heavily contaminated with a potent drug. That residue isn't going to smoothly and evenly mix into the entire giant batch of placebo powder.
It's not going to just perfectly disperse.
Right. It is most likely going to scrape off and concentrate heavily in the very initial discharge portion of the batch.
So if the quality control team happens to take their sample from the middle or the end of the batch,
they will miss the contamination entirely.
And if the contaminant is a larger particle size, it won't disperse evenly either. It will just clump.
Exactly. The other major flaw is the dilution effect. By running a massive placebo batch through the system, you are vastly diluting whatever small amount of contaminant might be stuck in there.
You're just hiving it in more powder.
You are severely reducing the analytical power of your laboratory tests. Because of these problems, the FDA explicitly states that the placebo method cannot be used alone to validate a cleaning process.
It must be used in conjunction with rinse or swab samples. Which brings us to the actual cleaning agents used in those rinses. To get these machines clean, factories use heavy-duty industrial detergents and soaps.
They need the strong stuff.
But wait, if you use detergent to clean the machine, Doesn't that mean the detergent itself becomes a chemical residue that you now have to test for?
It creates a massive puzzle known as the detergent dilemma.
A detergent dilemma.
Yes. Product residues from the drugs themselves usually have logical, established safety limits based on medical science. The industry might use limits like 10 parts per million.
Or 11,000th of a normal therapeutic dose.
Exactly, to determine what is an acceptable level of leftover active ingredient.
10 parts per million. To visualize that for you listening, imagine a standard bathtub full of water. 10 parts per million is roughly just a few drops of water in that entire tub.
It is an incredibly tiny amount.
But we tolerate those tiny drops for previous medicines because we know exactly what they do to the body. Detergents are a totally different story.
Detergents are not medicine. They are not part of the manufacturing process. They are only added to facilitate cleaning.
Right.
Therefore, the FDA expects that absolutely no detergent, or at least very, very low levels for ultrasensitive tests, remains after cleaning.
They must be completely and easily removable.
Totally gone.
But the text notes a huge, frustrating problem with this requirement. Detergent suppliers often hide their specific chemical compositions as trade secrets.
They won't tell you what's in the soap.
So if the pharmaceutical company doesn't know exactly what chemicals are in the industrial soap they just bought, how can their lab accurately test to see if the soap has been completely washed away?
Often, they can't.
Wow.
And the FDA's stance on this is unwavering.
Yeah.
You cannot evaluate the detergent residues because the composition is secret, or if the detergent is chemically formulated in a way that makes it too hard to wash out of your specific equipment, you have to select a different detergent.
The manufacturer is fully responsible for proving the soap is gone.
No excuses.
The FDA really doesn't leave wiggle room. Which leads us to one of the biggest, most dangerous pitfalls mentioned in the text.
The test until clean fallacy.
Yes.
This is a stern warning from the FDA to their investigators to watch out for a very specific type of bad behavior.
What are they doing?
Some manufacturers utilize a concept where they clean the equipment according to their standard process, test it, find unacceptable residue, clean it again, resample, and retest until they finally manage to get a single passing result.
And then they just use that one pass?
They point to that final passing grade and claim to the FDA that their standard cleaning process is validated.
That completely defeats the purpose of validation.
Completely.
Validation is supposed to prove that your standard operating procedure consistently does what it is expected to do on the first try.
It is like taking a driving test over and over, crashing the car into a wall 5 times, passing on the 6th try, and claiming you are a perfect validated driver.
It just doesn't work that way when human lives are on the line.
The FDA text makes it incredibly clear. For a system with a validated cleaning process, this practice of constant resampling and retesting should never be utilized.
Never.
In fact, if a company is constantly retesting, it doesn't prove their process works. It actively documents the exact opposite.
Because they keep failing.
It proves on paper that their standard cleaning process is wildly ineffective, leaving unacceptable residues and contaminants behind on the initial pass.
We have covered so much ground today. We have explored the hidden, microscopic science that goes into ensuring the medicine you take is actually pure.
From the shocking history of pesticide contamination caused by reused drums,
To the battle against stagnant water and fever-inducing pyrogens.
we've seen how something as simple as the adhesive on a cotton swab can derail a validation study.
And why running a placebo batch, or just testing until it's clean, are dangerous illusions of safety. It is a massive, relentless undertaking to guarantee that clean actually means clean.
This raises an important question, especially when we look toward the future of pharmaceutical manufacturing.
Where is this all heading?
The text points out that with advances in analytical technology, we can detect residues at incredibly low levels. Those detection capabilities dictate the limits we set.
Like that bathtub analogy of 10 parts per million.
Exactly. But analytical technology is advancing exponentially. We are reaching a point where we can detect single molecules of a substance left behind in a 1,000-gallon stainless steel vat.
Single molecules.
If our ability to detect contaminants becomes infinitely sensitive, will the pharmaceutical industry eventually face a paradox?
What kind of paradox?
Will zero residue become technologically impossible to prove simply because our sensors will always find one stray molecule?
Wow.
We may eventually be forced to completely redefine on a philosophical and scientific level what the word clean actually means.
That is a fascinating thought to end on. A future where our microscopes are so good, nothing can ever be legally clean again.
It's something to chew on.
Thank you so much for joining us on this deep dive into the invisible world of pharmaceutical validation. We hope you walk away with a newfound appreciation for the incredible science protecting your medicine cabinet. Keep asking questions, keep exploring, and keep learning.